Hello everybody, my name is George James and I'm an assistant professor with International Hellenic University in Greece, but also the deputy chair of the UNESCO Chair Connect. Today in this video we will be presenting to you some methods to monitor and assess riparian areas and deltas. I'd like to thank very much my co-authors, who you can see here in the pictures, Alasia Kovoglu, Georgios Giatas, and Georgios Pagonis. So, in this video, we will start with what a riparian area in a delta is. Then we will present the methods we believe that you need to start assessing or monitoring these ecosystems. These are called the preliminary analysis methods. There's two main categories, protocols and GIS. The next step is to go into methods that are more specific for a reach or a catchment where we have diagnosed a problem. In this case, we have small scale and large scale methods. After the description of all the methods, we will tell you our thoughts, what we think the advantages or the disadvantages of each method is, and in case you want to utilize and learn more about these methods, we also provide you with references. So, when you go next to a stream or a river, or in general a water body, you can find these riparian areas. They're different from the adjacent ecosystem, and this is because of the excess water they have, firstly from groundwater, but also from the flood events that typically occur in these areas. These lead to unique soils and also unique vegetation that is typically hydrophilic. They call these transition zones between aquatic and terrestrial ecosystems, and they're very important because they offer many ecosystem services. Deltas are geomorphologic formations. You can see them here in this picture in the yellow circle. They are formed as a river or stream enters a slower water moving body, like a lake, sea, and ocean. So this is an extension of land that is created by the sediment that is deposited. It's also very unique. It's also considered a transition zone, offers many ecosystem services, and the vegetation in many cases is riparian. Both these ecosystems are very important in water-scarce regions such as the Mediterranean. In the Mediterranean, they have played a very important role in the welfare of the people, but also for the development of the civilizations. In addition, in this region, we can find them along different types of water bodies. For example, riparian areas can be found on intermittent or ephemeral streams, perennial streams, small, medium, large size rivers, and also deltas. But they offer many ecosystem services, have been heavily utilized, and need to be protected and conserved. We are also very interested in promoting nature-based solutions. According to the IUCN, these are actions to protect, sustainably manage, and restore natural or modified ecosystems, while at the same time providing human welfare and biodiversity benefits. Why do we like these solutions? Well, they may take longer to be accomplished, larger areas to be implemented, but in the long term, we believe they provide more sustainable and more realistic solutions because they are based on ecosystem and environmental principles. So let's start with the preliminary analysis methods. We have the large-scale methods that are based on GIS, and specifically a GIS function, the buffer, so we can look at riparian areas at very large scales. And then we have these that are at a smaller scale, for example, a reach. And in this case, we have used visual protocols. Typically, we use the visual protocols to validate the data we have based on GIS. And after that, using both of these, you can find, for example, reaches or catchments where we can implement targeted approaches. So let's now start with the large-scale methodology for the preliminary analysis, which is the GIS. One of the reasons we recommend these because most services today have an expert in GIS or have a lot of personnel that know how to use GIS. The first step, of course, is to download satellite images or maps that you have, and then digitize the stream network and the watershed boundaries. In many cases, this information is already there. Then what you do is use a function in GIS, which is called the buffer function, and you can see this in the middle picture, where along each side of the stream, you can see what the land uses are. Now, because, as we said previously, you can have riparian areas along streams, small rivers, large rivers. You can choose the buffer size you can have. It can be, for example, 5 meters, 10 meters, even up to 50 meters. And this is what you can see on the picture on the right. And here's a more specific example. So we were interested in this reach. We digitized the stream, and then we took the buffer. And then what we did is we also digitized the different land uses that were in the riparian buffer. It's very important to know the land use in the riparian area because it's different to have a natural forest or urbanized area or, for example, a cultivated area. So now let's move on to the preliminary analysis methods for the small scale, specifically protocols. We primarily use the stream visual assessment protocols, or ASVAP, that focuses on the stream channel. Since this was developed by the NRCS in the United States, we have modified it in order to be better adapted for the Greek riparian areas. What you do have is 14 different characteristics that can get values from 0 to 10. 0 is the minimum. And then you have 4 different categories. If the average of the above 14 characteristics is above 9, you're in excellent condition. If it's below 6, it's in poor condition, and you need to start thinking about potential management measures you need to take. Of course, you have the 2 intermediate categories of good and moderate. This is the typical field sheet you need to complete. The first one has general information about the site. A lot of this can even be completed in the office. And you also have some space in order to draw the exact location where you took your measurements. The second sheet, as you can see, has the area where we will put the values for the 14 different characteristics. You also have some place in order to put some notes if you think these are necessary. So this is the end result. This is one of the reaches that we implemented the stream visual assessment protocol. We had 20 different sampling points, and what you can see, most of the sub-reaches, especially in the middle and downstream reaches, were in moderate condition. We had 1 in good condition, none in excellent, which is really worrisome. And then in the upstream reaches, we had 4 that were in poor condition. So from this map, it tells us if we want to do some targeted approaches, we should focus on the upstream reaches. The other protocol that we really like is called the Ecological Quality of Riparian Habitat. The acronym is QBR, and this focuses more on the riparian vegetation. It has 4 different sections, and each of these looks into different characteristics. The first 3 are on the riparian vegetation, for example, if you have fragmentation, if you have different canopies. The last one focuses on the stream. Each section can get a maximum of 25 points, and the maximum score you can get is 100 points. If you have 95 or greater, you're in very good condition. If you have below 25, you should be really worried. Of course, we have the intermediate categories with good, fair, and poor quality. This is a typical form that you need to complete. It's a little bit more complicated than SVAP, but still, with a little training, we found out that you really can use it, you really can understand it if you have a basic knowledge about watersheds and riparian areas. So once you have conducted the preliminary analysis methods, then you can identify specific reaches or catchments that you want to focus on. And here you can implement some targeted approaches. For example, at the small scale, you can use different methods to measure stream flow. You can also assess water quality. And what we're lately dealing with is using bioindicators to tell us the quality of the riparian habitat. Now, if it's for an entire catchment, then you also need to use some new technology methods like vegetation indices that are a remote sensing technique, but also models. We've used two types of models, hydrologic models and prediction models. So let's start with the most basic. You have a stream, you need to know what the hydrologic regime is. Especially in the Mediterranean, you'd like to know if it's a perennial stream, intermittent stream, ephemeral stream. You'd also like to know when the peak flows are, so you know when there's a potential for flooding, but also when the low flows or no flows are, because in that case you might have some problem with drought. Ideally, you'd like continuous measurements. Of course, this is quite difficult. In our case, we've used primarily the current meter because we usually assess small streams. And in regard to the frequency, we recommend this will depend on your objectives and, of course, the time and funds you have for the specific project. So, you can measure the stream flow at one location or multiple locations. At the specific location, what you do is measure your cross-section, and then you can separate this into different subsections. In each subsection, you measure the width, the depth that gives you the area. In addition, you go at 0.6 of your depth using the current meter and measure the velocity. Typically, we like to measure velocity 3 times. Then you multiply the mean velocity by the area to get the discharger for the specific subsection. By adding all the discharges for each subsection, you can get the discharge for the specific cross-section. So now we have a small video to illustrate how we conduct the stream flow measurements. So here is a typical Mediterranean stream. This is after a precipitation event, and you can see that the water is flowing. This actually is from the Nestor's River, where I have one of my graduates taking the stream flow measurements. You can see that the propeller is not going very fast, so we are not having very fast flow. I think the other nice thing is that we were able to capture in these images some of the small fishes. Now we move on to a much smaller stream. This is a Ia Varvara stream in Drama City, and you can see the water is moving much faster. Of course, the water can move even faster. Now, in regard to water quality, it's also important to know what is going on with your stream. This will give you information on potential pollutants. The frequency, ideally you want very frequent measurements. Of course, this will depend on the objectives of the study, it will depend on the funding you have, and depend on the time you have. What we've measured in the past is temperature, pH, dissolved oxygen, conductivity and alkalinity, suspended sediments, turbidity, ammonium, nitrates and nitrites, orthophosphate, total phosphorus, total coliforms, and E. coli. We thought it would be nice to also show you another video. So this is some of the equipment we use in order to take some measurements in the field. This is one of my graduate students taking a water sample. Part of the water sample goes into a plastic tube where we then do the analysis in the laboratory, but we can actually do some of the analysis in the field. For example, temperature, alkalinity, pH, conductivity are some of the things that we can measure in the field. We're looking also into utilizing bioindicators. One of the reasons you prefer these is because they're much less variable compared to physical-chemical parameters of water that you measure. Now, if you find the appropriate species, then you can actually detect habitat changes due to anthropogenic impacts. Of course, the key is to find the proper species. You cannot use rare species, and you cannot use ubiquitous species. In many cases, what we've done is we've measured the entire population, and then we've utilized diversity indices. Specifically, we used Shannon-Weiner Diversity Index, Shannon-Weiner Equitability Index, and the Simpsons Diversity Index to compare different sites to see if different habitats have an impact on these bioindicators. The first bioindicator that we experimented with was small birds. Sometimes they're difficult to see, and of course you need somebody with experience in identifying them. But they are very sensible to changes in riparian vegetation and riparian structure. What we did is at specific locations along a 50-meter diameter area, we counted all the birds we can see and can hear. In addition, in many cases, what we do is look for certain characteristics. For example, if there's any nests of birds or if there's signs of bird activity on trunks. We've also used ground-dwelling insects. In this case, in order to monitor them, you use pitfall traps. You can see pitfall traps in the first and second photo. Then the second photo are the actual pitfall traps that we used in the field. So what happens with these traps? Ground-dwelling insects fall into them, and then because we have a certain fluid, like an antifreeze, it kills them and they are maintained. So after that, you go and you measure how many different ground-dwelling insects you had and also their populations. In many cases, because it's difficult to measure the entire populations, you can focus on a specific species. So, for example, in our case, we used Sipha obscura to compare different riparian habitats. And you can see a Sipha obscura on the right in this slide. We've also looked into dragonflies. Greece has many different and beautiful dragonflies. So what you do initially is you collect the different species you can find in a specific riparian area, and then you kind of familiarize yourself with them. After you do this, you go to the site for specific periods in time and just count how many different species you see and their number. So now let's move on to methods that can be used at much larger scales. We will start with hydrologic models. Nowadays, these are used worldwide. They can be very effective, especially at large scales like watershed or the basin. We've primarily worked with the soil and water assessment tool. The reason is because they can predict the impact of land management practices on water, sediment, and chemical yields. And they can do this in large complex watersheds with different soils, land use, and management conditions. And of course, this can be done over long periods of time. And here are some of the results that you can get from SWOT. In this specific case, we were able to get the hydrologic water budget that told us how much of the precipitation water will be evapotranspiration, how much groundwater flow, how much surface runoff, and how much lateral flow. I like to say at this point that the validity or the results of the model depend on your input data. If you have good input data, then you'll have good output data. So you need to have good information on your land uses, on the topography, on the soils, and also streamflow data in order to be able to validate your model. The other large-scale method that we really like are vegetation indices. These are based on remote sensing techniques. Specifically, you look at the reflectance of the vegetation, and depending on this, you can see if the riparian vegetation is in good or bad condition. In addition, another advantage is that we have now satellite images for many decades. So actually, you can look if vegetation is changing through time. In our case, we use primarily the following indices, specifically the Normalized Difference Vegetation Index, NDVI, the Normalized Difference Water Index, NDWI, and the Land Surface Vegetation Index, LSWI, because we have focused most of our research in riparian areas. So what do you do here? You have your satellite image, then based on an algorithm, you can actually even do it in GIS, you come up with a map as the one you see on the right hand. Here, anything in green means that we have good vegetation, anything in red means that we have poor vegetation. Now this is for the entire watershed. In some cases, we want to focus only on the riparian area, as you can see in the image in the middle, and there you can find out what is going on with vegetation in your riparian areas. Here is another study that we utilized vegetation indices. The study area was the Nestos Delta, and we had satellite images starting from 1989 till 2018. What we did in these images, we implemented 8 different vegetation indices that you can see in this figure. Now, in most of these figures, we can see that there is a decreasing trend. That also tells us that the riparian vegetation quality is decreasing. So this should worry the people that are in charge of the Nestos Delta. And I think we can attribute this to the fact that after 1989, 2 dams were built in this specific river. Finally, in regard to large-scale methods, we're also using prediction models. Specifically, we are using the random forest prediction model. We have implemented this in R, and one of the reasons we like this is because it can handle heterogeneous data that originate from different sources and have different scales. In addition to that, in the end, it gives you some metrics that can tell you the importance of the different predictor variables that you have used in regard to the variable that you are interested in. In most cases, we are interested in the riparian vegetation. So we implemented this method in the Nestos Delta. Specifically, we wanted to see if anything is going on, or if we can find more specifically any trends in regard to the riparian vegetation. So what we did, we compared the values of the 8 different vegetation indices we had with what we call predictor variables to see which of these impact more the vegetation. We had 26 different predictor variables. Some of them were territorial, like distance to the sea, distance to the river, distance to the dam, and the other were climatic ones, like mean temperature or yearly precipitation. The results are quite easily to interpret because the greater the value one of the predictor variables has, the more important it is. And what we found was, at least in this case, that distance to sea, distance to river, and distance to dam were more important. So, what are our conclusions based on these different methods that we have utilized? We like to start with GIS. It will really provide you a quick assessment. It will tell you what the land uses are in the riparian area. So it provides you a really good first perspective of what is going on in the stream or in the channel, in the stream channel. The next step for the preliminary analysis is to use visual protocols. We've tested and experimented a number of different visual protocols, and our preference is SVAP and QBR. The reasons are they can be easily and quickly applied, and they're also highly repeatable. We've had students of ours in different classes do the protocols on the same reaches and find similar results. Finally, we like to use both of them because they're complementary to each other. SVAP focuses on the stream, QBR focuses on the riparian vegetation. So we found that for the same specific reach, very different values based on the different protocol. After you do the preliminary analysis, you can go into specific reaches and watersheds where you want to focus on. In these specific reaches, it's important to know the hydrologic regime. You can either do this with a current meter like we've been doing, or if you have more money, you can have a hydrometric station and take more continuous measurements. I also believe it's good to get some water quality measurement, especially if the stream water is used, for example, for households. Now, another advantage is if you get some water quality measure to understand what are the main pollutants, and if you know the main pollutants, then you can find the resources and recommend some management practices to reduce these pollutants. Finally, we found that bioindicators are promising. Compared to the other 2 methods, they're more cost effective. You need to take less measurements. Of course, you need more trained people that need to either identify the birds or the insects. So we are in the 21st century. We need to start utilizing these new technologies that are out there. Hydrologic models can give you information about stream flow, about suspended sediment, about other pollutants at the watershed scale. Random forests can pinpoint at what are the important variables for the riparian vegetation. The indices can provide trends. What is going on with your riparian vegetation? Is it improving or is it degrading? Compared to the traditional now site assessment methods that are very laborious, resource demanding, and time consuming. These methods actually can be applied at a much wider scale. We can have longer term data sets. We can also have a large number of data points. But we need to notice that field measurements are required. These will calibrate and validate the results from these new methods. Here now we will present to you some of the references in case you'd like to do similar research. And the first reference is about methods you can use for assessing riparian areas. The second one refers to climate change impacts on Mediterranean riparian areas. And we also have a case study of urban riparian areas. We move on to a study on nature-based solutions. And the last two are the reference for the SVAP and the QBR. Now, as we said, we have modified the SVAP. So the first two references can give you some information about the modified SVAP. And the last three references are about the bioindicators we have used. Small birds, the ground-dwelling insects, and the dragonflies. So in this last slide with references, we start with a reference on how to use GIS to assess riparian areas at the watershed scale. We continue with two references that describe how we use the vegetation indices, random forest, and SWOT. Then we have a recent case study that we are doing with the municipality of Drama, and the last two references are general about riparian areas and studies that have been conducted on riparian areas in Greece. At this point, I'd like to thank you very much for listening. I hope you learned something from the video. I hope it was entertaining. Here is my contact info. In the end, you can see my email. Email us if you want more information about the studies we've done. Email us if you want to maybe collaborate and do similar research in your area, and maybe we can have some research going on together. Again, thank you very much for listening.